Okay, welcome to Roasted Academy. So, let's talk about the complement system. The complement system is simply a set of proteins that is secreted by the liver, and secreted by the liver, whereby these proteins are inactive. Okay, these are inactive proteins, but when there's a presence of pathogens, they become active. They become active and they help in inflammatory reactions. They try to get rid of the pathogens. So these simply are a group of about 30 proteins which augment or complement the immune system. So most of these proteins are released by the liver, then they are secreted in serum. So like I said, these are synthesized in the liver as inactive precursors and are activated by proteolysis during their interaction in a sequential manner. So one of the protein will activate the other protein, the other one will activate the other one. That's how it is, right? Yeah. So there are three main functions of the complement system. Lysis of the cells, okay? Generation of mediators of inflammation, opsonization. So we're going to see that in the end, there's what we call as the membrane attack complex, which is going to cause some holes on the bacteria. As a result, there's going to be leakage of the intracellular substances of the bacteria. Then the bacteria would die, the pathogen is going to die. That's how the complement system gets rid of the pathogens. Generation of mediators of inflammation. There are some inflammatory molecules that are generated by the complement system that we mention. There's also opsonization, which is the enhancement of phagocytosis. So you are going to find that in the complement system, the antibody binds to the pathogen, then it gets activated, and the FC portion will attract the macrophages, for instance, to come and phagocytose the bacteria. That's one of the practical example that we see in terms of enhancing phagocytosis. So there are over 30 serum anti self-surface proteins. That's the complement system. So we've got three pathways of the complement system. I've got the classical pathway, the alternative, and the lectin pathway. The classical pathway, it simply is antibody dependent pathway and is going to be triggered by soluble antigen antibody complex. So when there's antigen antibody complex, that's when this particular pathway is going to be triggered. Or by binding on the antibody to the antigen, when the antibody binds the antigen, then so this one is antigen antibody complex activated. Then the second one simply alternative. Alternative, we simply mean that it's the alternative of the classical pathway. There's no antibody used here. Okay. So no wonder they're saying is antibody independent pathway, which is simulated by the antigens direct. So it can either be bacterial surface components. Okay. Then we go to the lectin pathway, which is also a type of pathway that is antibody independent. Okay. This one is simply stimulated by the presence of the mannose on the pathogen. This one gets activated. Okay. So now, in looking at the complement system, we look at each of these pathways and how it's going to get rid of the infections. We look at the classical pathway, we look at the alternative pathway, we look at the lectin pathway. Three main stages in the activation of the complement system by any pathway are: 1. We form the C3 convertase. Okay? When we mean C3 convertase, we simply mean that the one that is going to cleave C3 into C3A and C3B. Formation of C5, again, we say C5A and C5B. In the end, we also have formation of the membrane attack complex. Okay? So, thank you so much. In the next video, I'm going to be talking about the classical pathway, how it takes place in the end, right? Alternative as well as the lectin pathway.